Hi, hello, this is Paulo Rebecca from BIOS. I'm gonna talk about the genomics use case, and in particular, I'm gonna illustrate this application that we have, which is the porting of a genomic alignment software to a Phasm. I'm just gonna give a brief introduction, then after me, Simon from Imperial College will give a demo and more information about the porting. Essentially, well, we can say that we are now living in the age of genomics. Genomics, which is the study of sequences, biological sequences, as DNA or RNA, nucleic acids in our cells, are now essentially in a number of applications. You can think about cancer genomics, personalized medicine, or of course, recently COVID-19. So this figure just shows you a phylogenetic tree which has been reconstructed out of the sequences of thousands of COVID-19, of SARS-CoV-2, as the name of the virus in fact is, sequenced during the first stages of the pandemic. Of course, you could not have any vaccine, any hope of a cure without this kind of genetic information. And of course, the idea is that viral sequences are just a small minority of the sequences which are available worldwide. If you just take, for instance, SRA, which is one of the repositories where this kind of genetic data is submitted, you can observe that increase, in fact, is essentially exponential, and several petabytes per year get added to this kind of data repositories. That can be understood if you think that one single sequencing experiment can produce up to several terabytes of data in one single run, so in a few days. So essentially, of course, this opens a lot of problems as for, in particular, processing of this data. Of course, it is now impossible for every institute to store a copy of this data, so we all rely on this kind of worldwide shared repositories. And then, of course, it's also impossible to reprocess all of this data as a single institution. So what you need, essentially, have to rely on the cloud if you just want to temporarily take some of this data and reprocess it. However, computation in the cloud is very expensive, and it's very difficult to make things scale as you would like to for this kind of data. So that opens the way to serverless computing, which offers the possibility, at least in principle, to essentially have cost-effective computations on the cloud that scale much better than what we have at the moment. So essentially what we thought for this genomic use case was to try and port to serverless platforms some kind of data processing application. So in the jargon, we call this kind of data analysis workflows pipelines, because information flows through a computation graph, and each block represents a very complex, in general, application that just processes the data and returns a step of the computation. As we're saying, each step can be, in fact, made of like thousands of tens of thousands of lines of some programming language such as C, C++, or OCaml, which is a functional programming language that we like to use very much because it's very suitable for bioinformatics applications. It's very concise but very powerful for parsing and other applications. So we have this kind of computational pipelines which are high throughput and essentially allow bioinformaticians to process a lot of genomic data in a short amount of time. And what we need to do for this kind of applications is run the data through a series of steps, which can be very complicated depending on the application. But essentially, the basic problem is that the sequencing machines produce short reads, which are strings of up, let's say, to 300 letters. Each letter can be a DNA base, so A, C, G, or T. And then what happens is that those strings can be somehow found in the reference genome for humans. So essentially, a few years ago, at the beginning of the 2000s, the human genome has been sequenced, and although different individuals have slightly different genomes, it's still possible to have a reference genome for the human species, which means that it is not exactly the genome of any single individual, because the genomes of every single individual differ from one another. But essentially, it's sufficiently close to all the human genomes that it can be used as reference. The reference human genome is very large, is a string of about 3 gigs of ACGT letters. And then the idea is that sequencing machines cannot really decipher, decode the genome as just a single chunk, but return small chunks, which are about 300 letters each. And then what one has to do as one of those steps, which is called alignment, essentially one has to locate the small string in the original reference for the human genome, and so as to try and understand where this small chunk comes from in the big original genome. And essentially, in order to do that, you have to do a search with mismatches, because again, there can be sequencing errors, there are differences between reference and individual. So I have to search for this string in a 3 gig database, and you have to do it very quickly, because essentially you have to process billions of strings. So each one of those experiments produces like billions of strings, and then essentially you have to do a search for each one of them with errors. Then once you have located the short reads, so each of those very small segments would be a short read of 300, the idea is that essentially they would accumulate in places which are relevant to biology, depending on the experiment that you are performing, of course. But in general, you're interested in locating those because locating them returns you relevant information about biology which is going on. So we have developed, let's say, several years ago, as part of this pipeline, a novel algorithmic approach to this kind of alignment. So this kind of similarity search, quick similarity searches with mismatches in the context of DNA, so this ACGT genome. So it's optimized for doing the similarity search very quickly, this kind of short strings with an alphabet of just those 4 letters, essentially, up to a level of error which is compatible with the biology. So we published this work a few years ago, and again, it offers a lot of novelty. It's still today, I think, one of the fastest algorithms to solve a problem, and it has a lot, again, of technical advantages. But okay, I will not go into that right now because it's probably not interesting. What is interesting, though, is the fact that this stage essentially is implemented as like probably, I don't know, several tens of thousands of lines of C code. So it's a very complex piece of software, which is optimized for essentially multi-threading, so to be run on a server node, so on a cluster node. And again, it is optimized as for reading and writing so that it can provide a suitable throughput to process a lot of sequencing reads in a short amount of time. Now, the idea is that, of course, we'd like to port this to serverless computing in as straightforward and not painful way as possible. And on top of that, we'd like to make it scalable so that we're not limited to number of threads or CPUs, let's say, which are available on a single node of a cluster. The problem is that, again, this algorithm is designed with a node, a physical computing node in mind. And so essentially it requires a large index, as it is called in jargon, to be loaded into memory. An index is a transformed binary encoded version of the reference genome for human, for instance. And this, again, is so the sequence, the original sequence is transformed in such a way that once you load this transformed version into memory, you're able to perform exact searches of substrings very quickly. The problem is that, again, this requires to load in memory several gigs of data, and usually these amounts of RAM are not available to the serverless architecture, so framework. So this is one first problem that, again, any port into serverless has to tackle. And okay, now I'm just gonna give the floor to Simon, who will explain how this problem, how we have tackled this problem, how we have tried to solve it, and also how easy it is or not to port this kind of very complex pieces of software to serverless with Phasm.
Hi, I'm Simon Chilliker. I'm going to give a quick demo of the Genomics Mapping use case with Phasm. So the problem of genomics mapping is part of the larger genomics pipeline, and what we're trying to do is map a load of reads data to a larger reference genome. Now the reads, as you can see on the left, is just made up of A's, T's, C's and G's, as is the index on the right. So it's a little bit like an approximate text search. So in order to parallelize this, which is obviously our end goal, we need to split up the reads data on the left and split up the index data on the right. And this makes the algorithm itself easy to parallelize. So we can run this on a single machine and it'll look a little bit like this, where we have two threads on this one machine. The one on the left is looking at 3 chunks of read data and one of the chunks of the index, and the second thread is looking at the same chunks of read data with a different chunk of the index. Now each one will output one blob of output per read index pair. So you can see at the bottom there's 6 there. These live on the shared file system. Obviously, by definition, the scaling of a single machine approach is limited to the size of the machine itself. However, obviously single machine multi-threaded code is simple to write and easy to understand. So if we want to distribute this problem, we can do a similar thing, but have a thread on each of the machines handling one chunk of read data and one chunk of index data. Here we can see 6 threads spread across 2 machines, and each one will produce the same bit of output, one piece of output per read index pair. But because they're on multiple hosts, this output will need to live in some shared state, some shared storage like a database somewhere. So this is great because it scales to potentially hundreds of machines, thus speeding up our mapping. However, as with any distributed system, this is difficult to build and even more difficult to integrate with the genomics pipeline itself. And this is where the idea of serverless computing comes in. We can think of each of these threads as a serverless function, that takes in some read data and some index data and writes this output. And so to do this as part of Cloud Button, we took the Gem3 mapper, which is an open source mapping application, and we ported this to Phasm, our serverless runtime. And this gives us really simple but distributed mapping, so it's the best of both worlds. The porting effort is fairly minimal, as is the stated aim of Cloud Button. We took a normal multi-threaded C code base, i.e. Gem3 mapper. Admittedly, we had to add about 60 lines of new C code, but this is mostly just wrapping the existing application in Phasm functions. And so now we'll go on to a demo of how this works. Okay, so now we're going to just do a quick demo of the normal single machine code running on a single host with about 20 cores. So this is the first case that I outlined in the slides, where we're limited by the capacity of this machine, which, as I say, has 20 cores. So we should be able to go up to about 20 parallel workers. So first of all, we can look at the data, which, as we see here, is just these chunks of index data and chunks of read data, as I outlined in the slides. Now they make up about 14 gigabytes in total, where the reads data is around 2.2 megabytes per file, and then the index data kind of varies. We can see some of the files are a few hundred megabytes, some of them are over a gigabyte. So we can run the normal Gem Mapper application as we would in a non-serverless context, passing in a given reads file and a given index file. And we see that this can map that given set of reads to that given index chunk in around a second. So now we can run a full native indexing process by running all of our index chunks against all of our reads, but with varying numbers of threads to show how that changes the performance. So we can run this script here, passing in number of threads equals 10. And we can see that this will churn away, taking each read and mapping it with each index chunk. Now, obviously on a 20-core machine, we're using about half the capacity of the machine, so we can definitely get faster than however long this takes. But there's this limit. So we can sort of say maybe 20 threads is probably the maximum. So this is run in about 22 seconds with 10 threads. If we now run it with 20 threads, we are kind of maxing out this machine, and we'd expect it to run in roughly half that time. So yeah, 12-ish seconds. And then we can see that this will produce an output around 222 megabytes, and whoops. Yeah, as we can see, this output consists of each read chunk paired with each index chunk. Now these outputs will obviously be the input to the next stage of the pipeline, which will merge them all back together. So now we've done that native version, we'll do a demo of running that with Phasm. Okay, so now that we've seen the code running natively, as it would on a normal single machine deployment, we can look at running the code with Phasm. So the major difference when running with Phasm is that obviously we need to upload the read and index files beforehand. And we can just do this with the Phasm command line, uploading one of our read files here. And we can see that uploads fairly quickly, but obviously to do the whole 14 gigabytes of input data takes a little bit more time than we have here. However, once that's been uploaded, you can run the mapping in parallel as serverless functions with this function. And so in the background, this is spawning a load of Phasm serverless functions, which are performing the mapping in parallel, as described in the slides. And the key thing here is that we've not had to actually engineer much of the distributed system ourselves, other than writing the function, which is the whole aim of serverless computing in the first place. I've just written the code wrapping up the normal Gem3 mapper. And so although this is executing now on 3 hosts, each of which have 20 cores, the performance is actually similar to when we're running with 10 cores on a single machine. And this is one of the major limitations of our current implementation. So the Gem3 mapper code makes heavy use of some optimizations to access files on the local file system. Whereas in Phasm, of course, we are running in a distributed environment where the data is accessed from Phasm's shared state. Coming up, we'll be doing some optimization to make sure that these file system accesses are much quicker than accessing the code normally with Gem3 mapper. And then hopefully we'll be able to see this increased parallelism translate into lower running times. So just to dig a bit deeper into the scaling aspect of Phasm, here we see an execution graph of a different genomics mapping run. And this is what Phasm generates internally when it's performing one of these mappings. So we see at the top there is an initial kickoff function, which then spawns these 24 other functions which map the given read chunk onto the 24 chunks of our index. If we zoom in on the middle here, we can see each of these boxes gives us the details of the given function execution, including its running time and the host on which it was executed. Now in this case, each of the boxes has been colored depending on which host it executed on. So you can see there's 3 different hosts in this case, the green, the blue and the yellow. But obviously as we scale up Phasm and a given deployment, we can achieve much higher scalability, lots more different colors and lots more boxes. So as I mentioned before, the key part of our next steps is improving the performance of our current implementation. This mainly centers around improving the efficiency of the file system access that is performed with Gem3. Then we also have an opportunity to improve the Phasm scheduler to take advantage of the distribution of index chunks throughout the cluster. And then finally, we will look at integrating this with the rest of the genomics pipeline, obviously, and then parallelizing subsequent stages of the pipeline itself, such as the merging stage. Thank you.